Let me give a message. Okay? One moment. Muy buenas tardes. Vamos a dar inicio al curso ya este viernes y aprovecho para comentar sobre todo a los compañeros que nos están acompañando en streaming que el próximo martes no vamos a tener este servicio, pero vamos a ver la forma de transmitirlo por videoconferencia, por vía Zoom. Entonces, a todos los que estén conectados ahorita por Facebook o YouTube, les voy a pedir que por favor en el chat pongan su nombre completo y una dirección de correo para que nosotros les mandemos el enlace y puedan acompañarlos el martes, que sería la sesión que tendríamos vía Zoom. Los demás días de la semana sí tendremos el enlace por streaming, entonces nada más sería el martes, ¿no? Cuando cambiemos, digamos que la modalidad, y para eso necesitamos su nombre completo y su correo electrónico y vamos a estar tomando nota de estos datos durante toda la sesión. Entonces, sin más preámbulo, damos la bienvenida al profesor y comenzamos el curso. Muchas gracias.
Thank you very much. So I would say we start. I just need a presentation to get started, please. Now it works. Great. So we will start where we stopped yesterday. Maybe you thought a bit about a question, how we can perform a dark fermentation, how we can stop methanogenesis. Did you got any new ideas how we can achieve this? There's one idea or one thing that I mentioned already in the lecture. Maybe you got it. Okay, so does not seem the case. You want to say something? No? Okay, great. Then we will just go further and talk a bit about how to inhibit methanogenesis. So one thing is, which I told you already, that methanogenic archaea are extremely sensitive for pH. So we can just increase the pH, or it's actually much easier to decrease the pH, because as you know, doing anaerobic digestion, we produce a lot of acids, and the more of these acids are accumulating, the lower the pH will drop. And then when we are below 6, below 5, methanogens won't work so much anymore. So like this, we have methanogenesis already inhibited. But there are other options. For example, we can work with a very high organic loading rate and a low hydraulic retention time. So the high organic loading rate basically yields a low pH, because then we will have a fast acid production and the involved bacteria will produce the acids faster than the methanogenic archaea can consume the acids. And then the low HRT is related to the growth of the methanogenic archaea. They are growing quite slow. And this means when we have a very low hydraulic retention time, all the methanogens will leave the reactor before they have the ability to replicate themselves. So this is also a possibility to inhibit them. We can also use some chemical compounds, for example, coenzyme M analogues. Coenzyme M is one of the last enzymes or coenzymes involved doing methanogenesis, if we think about the methanogenic pathway. And this is a crucial enzyme, and if we inhibit it, then methanogenesis will be blocked. And there are some chemicals like lumacine that we can use, or bromethan sulfonate, or mercaptoethan sulfonate. But they are relatively expensive, so this is maybe useful for research, but certainly no one will apply this in the industry. At least not with large biogas plants or anaerobic digester plants. There are some other chemicals that we could use, but bacteria might be affected as well, so maybe it's not a good idea to rely fully on such chemicals. And there's a last option, we can perform heat shocks. So methanogenic archaea, they are also really sensitive to heat. And if you are heating up the whole system to 100 degrees, almost all the bacteria, all the archaea will die. And we have a lot of spore-forming bacteria who can survive this. So, for example, Clostridia. They form spores, and then once the heat shock is over, they can restart to grow. And then you have these bacteria there which can produce organic acids and also hydrogen, namely the main products of dark fermentation. Okay, I think now we have a... Yes, please.
I'm sorry, I have a question about the archaea. You mentioned that heat shocks will kill archaea, but we have already studied that archaea are
extremophiles.
Extremophiles. So how
yes.
could 100 degrees would kill them?
Yeah, so there are different kinds of extremophiles. Some of them survive heat very well. Others survive salt very well. Others can perform methanogenesis. But it's not that all of them have all the capabilities that we regard as extremophile capabilities. So methanogenic archaeas are usually, well, they die relatively fast when the temperature rises. There are some heat-adapted methanogens that you can find in the ocean, for example. But if we take the methanogens from cows, then they usually die relatively fast if you increase the temperature. The question is whether they could adapt. So let's say you have such an dark fermentation stage, and in the beginning it works quite well, but then maybe after half a year of application, suddenly they adapt to high temperatures. This could become a problem. But in this case, I would recommend to just combine several of these selection pressures. For example, you could have an irregular heat shock. I mean, you don't have the temperature that high all the time. So then it's more difficult to adapt. Maybe you have the temperature at 37 degrees, and then, I don't know, every 3 days or 4 days you make a heat shock. Or you have it as a batch, then you make a heat shock in the beginning, and then they die, and then that's it. And then if you combine this with a low pH, for example, then the chances are much lower that they will adapt to these conditions. But those are the ideas. I mean, researchers are still figuring this out, and so far I don't know any industrial plants where hydrogen is produced based on dark fermentation. So this is rather on a research level. There's one company that I know in Germany, it's the Snow Leopard company, and they have like a separated acidification stage, but they don't produce any hydrogen, or at least they don't collect it. And they don't produce organic acids either, or at least they don't extract them. So after the acidification is done, they pump the acids directly into the methane stage. And the idea there is to have a rather controlled process in order to prevent acidosis in the methane stage. So you can feed more and more acids, and then at the moment when you already see that the microbiome is a bit disturbed, and maybe the biogas plant stops increasing the biogas yield or even starts to decrease the biogas yield, then we can stop feeding more acids. And this works really well. For example, if we have a or one-stage digester, if you feed too much substrate and you see that the biogas plant is somehow disturbed, you can stop feeding, but the substrate will further acidify. So there's like a shift between stop feeding and stop acidifying. And you can prevent this shift if you have the separated acidification stage where already everything is acidified. So this is the concept of this Snow Leopard company, but they don't use it for biorefinery yet. So it's rather a pre-treatment for the pre-treatment of bio-waste. Okay. So I told you it's still a question of research and...
Sorry, I have a question.
Oh, yes, please.
Yeah. Sorry, I didn't understand how the coenzyme M works.
Well, it's an enzyme in the very last step of methanogenesis, but you have a wide range of enzymes that are all working closely together. So it's in the last step in this methanogenesis. There is this coenzyme M, and it helps to transfer the methyl group from the previous metabolite on another metabolite to form the methane in the end. it's just one of several steps doing the biochemistry of methanogenesis. Okay, so yeah, now we know how we could perform dark fermentation. We know how to stop methanogenesis, but we don't know how to extract the organic acids. Or maybe you do. Any ideas? How would you extract organic acids? No idea? You're shaking the head? Shaking the head? No idea either. Well, okay, I will present you some ideas. As I told you, it's all still part of research. So as we don't have these plants, no one is extracting it right now. But I mean, it's chemistry. So the methods for extracting these metabolites are no magic. So they're elaborated really well. But of course, there are some problems with the digestate itself. One problem is that we usually have a very thick sludge, which will block all kind of filters that we could apply. It's also not so nice for distillation, so it's not the best substance to perform an extraction. So there are 3 methods that I found in a recent review article. And we can talk here about solvent extraction, ion exchange and adsorption, and electrodialysis. I suppose you have heard these methods somewhere already in chemistry. I guess you had some chemistry studies doing your engineering studies. So let's start with the first one, the solvent extraction. The solvent extraction is also about distillation. But before we perform this distillation, we try to concentrate the acids more. And the idea is that we have like an oily phase, which is swimming on our liquids, on our digestate. And then we have some transfer molecules. So here's one of them. And this is something that we can call an amphiphilic amine. So it's a long chain, so it dissolves relatively good in an oily phase. But then you have also a polar group here. And organic acids, as you remember, they have a long hydrophobic tail and then a hydrophilic head group. And this hydrophilic head group could now bind to the polar part of this amphiphilic amine. And like this, it's pulled into this oily phase, which is swimming on top of our fermentation liquids. And like this, we can concentrate our organic acids in a smaller volume, and then it might be easier to extract it based on distillation. So in theory, this sounds relatively simple, but in this review article that I found here in regard to these 3 methods from Murali et al., they're discussing this, and yeah, they said that there are some problems. It's difficult to find a suitable chemical which is doing this very well. So there are several chemicals that are known, but not all of them are working fine. And also the pH is a problem. So usually we are working at a very low pH. So you need a substance which is also suitable to work at a low pH. And people are just trying different combinations. Maybe it's also a little bit empirically. And in this work from Murali et al., they are just giving some examples what has been used. So here you have always a combination of some oily substance like this oleoyl alcohol, for example. And then you have one aminic compound like this alamine 336 or adogene 283. And they all look kind of similar to this substance. So the idea is always that our organic acids are binding to this polar group, and then this whole complex is solving in our oily phase, which is swimming on top of our fermentation liquid. Okay, That's one option. Now we can come to the next one. Ion exchange and adsorption. So this is usually done within column. So this is supposed to show in column. And you have some kind of a matrix here. And then you have some ionic groups. And some of the ions can dissolve. So you could, for example, have here a lot of sodium, which you might wash away with your organic acids. So in the end, it's a basic chemical equilibrium. So you have all the ionic groups covered, and once you enter your organic acids, they are washing away the counter ions, and then they are attaching to the matrix. There are different matrices that you can actually use here. And in the end, you can wash it off again. So if you apply a lot of the counter ion, then you will wash out the acid in the end, and then you can collect it. So here are some compounds that you can use for this matrix. And well, in theory, this works well. But again, we have a problem when we are working with an anaerobic digester sludge. I don't know whether you have seen a sludge like this. Sometimes they are so thick that you can almost hold them in your hands. And if you try to pump something like this in a column, then it gets easily blocked. And you have a lot of other things which could attach to the surface of your matrix. So the efficiency would not be that good. So we made some experiments in our lab, and well, I was not too satisfied with the results, but yeah, it works. There's an interesting option which has a similar concept, but not in regard to ion exchange, but to adsorption. You could use biochar to wash your fermentation liquid. And the idea is that a biochar is really porous, and then some molecules might enter into these pores. So once you incubate your char for a while with your fermentation liquid, then you can try to separate the biochar, and then you can wash it. If you wash the biochar now with water, with deionized water was used in this case, then you can recover a large portion of your acetic acid. But there are other acids which are formed as well, and they are apparently retained in the biochar. And if you now wash the biochar again with ethanol, then you can wash off all the butyric acid. So this gives you the possibility to selectively take out some of the organic acids from your fermentation. But then in the end, it's not too clean, and you have to treat it again, I don't know, by distillation or some chemical methods to clean it from other substances. Yeah, and then you can also use electrodes to separate organic acids from your liquid. For example, here's a 3-chambered system, and then you have your acid going in all of these chambers, and then you're putting some voltage on your anode and the cathode, and then they will pull. They will pull the chemicals through these permeable walls or semi-permeable walls, and yeah, then you can enrich in one of the chamber the anion, and in the other chamber the cation, and then you can separately collect them. So like this, you can also concentrate your organic acid a bit more, but then again, you have to treat it again to get it in a really clean form. So I guess you would have to perform distillation again. So in all of these cases, there are some buts, and especially with this thick sludge, it's difficult. I can also imagine that's not too easy to handle such a thick sludge in such a cell. There are also some trials to improve these separation processes. For example, here the researchers were putting a polar wall inside such an charged chamber, and this improves the formation of ions, which facilitates then the separation of your organic acids. But in the end, I don't think it's the solution in order to make it attractive for the industry in the end. Okay, with this we can finish our lecture from last time, so there was not much left. And yeah, we started yesterday to talk about this work from Vainaina et al. They were giving a holistic overview on how you can perform dark fermentation. We learned about several products that we can produce, especially from the organic acids that are enriched there. They also talked about hydrogen. And yeah, so we can, for example, produce bioplastics, certain additives, lubricants. So there was a wide range of products that we could actually produce from these organic acids. And well, they also explained the case that we could use pure cultures. Pure cultures are, of course, well, they work, we know how to do it, but they are expensive to handle, and they are also prone to contaminations. So the idea with the dark fermentation is to make it in the mixed culture in order to get it to get it cheaper. But then we still have these technological problems on how to properly extract your products. Yeah, we also talked about several parameters that we can use to optimize the acid formation, like the pH, organic loading rate, hydraulic retention time, the temperature, and also the product concentrations. And we also talked about the importance of inhibiting methanogenesis, because otherwise all the organic acids that we're producing, as well as the hydrogen, will be consumed by our methanogenic archaea, and then in the end we have methane again. And now we heard about these three technologies for extraction of organic acids, so namely the solvent extraction, ion exchange with columns, charged columns, or adsorption-based methods with biochar, and also the possibility to perform an electrodialysis. Well, that's it. Are there any questions? No? Okay. Well, then we can move to the next presentation. Can you change the presentation, please? Thank you very much. Okay, so let's start with our next chapter. I made a small change, or actually it's not that small, but I told you that we are a bit short in time, and I was thinking about how to reduce it a bit, or to remove not that important information in order to make it possible that we finish all of the things until the end of this course. So we had one chapter about biochemistry of anaerobic digestion, and I was removing it, or a large part of it, and then the really important things I moved yesterday into this chapter of taxonomic profiles of anaerobic digestion. So we have one chapter less now, and we will now come directly to the taxonomic profiles. And we can now apply the things that you learned about sequencing. I hope you still remember how we perform sequencing. And now we will talk about what we can do with it, what results we can expect, and how we can analyze these results, how to make interpretations out of it. So here you see a content line for the things for today. So there's one article which was actually one of the first articles from my PhD thesis, where I analyzed 7 biogas plants in Germany. Then we will talk a little bit about the taxonomic ladder and unknown microorganisms. And maybe you remember these nanowires. We mentioned them already several times. The fact that some microorganisms are able to send electrons away using kind of nanowires which they are forming themselves. So we will catch up this idea today again, and we will talk about a topic which is called mediated interspecies electron transfer versus direct interspecies electron transfer. Maybe you have heard of it already. Then we will see another paper, which was also part of my thesis, of my PhD thesis several years ago, where I tried to simulate a multi-stage process and I tried to sequence the microorganisms in both stages. Then we will hear a little bit about thermophilic processes to complete the picture, because I did all my experiments at mesophilic conditions. And yeah, you will see that at thermophilic conditions, the results are just a bit different. And in the end, we will talk about robustness of taxonomic profiles. We already mentioned the robustness of taxonomic profiles when we talked about microbiomes and their capabilities. But now we will directly work with the robustness and get some deeper insight into this. So let's start. So this is now the article that I published relatively in the beginning of my PhD. So the letters are really small and you don't have to read them. It's just for you to read later on when you're learning the slides. I will explain everything what there is to explain. So it's really just for the reading at home. So let's start here. Do you remember what this was? Yeah, you're nodding, the 16S gene. I took again the one from Prevotella pallens, but there are many more. And I will talk again about the ribosome. So we know the ribosome already, and that's the 16S gene. Remember that I told you that there are some conserved regions which make the ribosome, or which are there because the ribosome is really important for our metabolism. Without the ribosome, we're not able to produce proteins, and we need proteins to build our cell structure and also to produce enzymes. So without ribosomes, no life is possible. Therefore, we have some really important positions in the 16S gene. The 16S gene is one of the RNA genes which are composing in the end the ribosome. Well, and all the green ones are conserved. They are not to be changed in order to not lose the functionality of the ribosome. And then we have the gray ones. So they can change. They apparently are not that important. If they are changed, then the ribosome can still be functional. And then we have mutation. So all kind of big bacteria are here. They are all mutating. And well, they are not mutating in the green regions. They are mutating in the gray regions. So we can try to find probes which are attaching to the green ones. Like this, we can specifically target the 16S gene. And then let's say we target here, and then we perform our polymerase chain reaction and all the parts for the sequencing that we discussed. So we sequence from there all the gene or a big part of the gene. And once we read the sequences, then we can also read the sequences which are very specific for certain microorganisms. Like this, we can distinguish them in the end. Is this clear? Yes? Okay, perfect. So I was applying this technology, and I did it in my home region in Thuringia. This is Thuringia. It's in the middle of Germany. It's the green center of Germany. And there is a city in the middle called Jena. Very often when people ask me where I'm from, I say from Jena, and then they say, oh, from Vienna. No, Vienna is in Austria, and Jena is in Germany. So I'm from Jena. So I was driving around, I was visiting places close to Jena, and I was closing places which have biogas plants, of course. And here you see all the names of the cities that I were visiting in order to take some samples. Here just some short names for each of these sludges, because otherwise it would be too much text. And I also highlighted different kind of digesters that we are analyzing here. So we have basically 3 kinds of sludge. We have very thick sludge from CSTR or plug flow. CSTR, you may remember, it's the continuous stirred tank reactor. Yeah, and this is so thick you can almost hold it in your hand. So it's really, it's not that liquid anymore. But they are still able to pump it. So they are at the border where you can still pump something. Then we have the leachate from these garage reactors, which you also have seen. And we had some sewage from the sewage sludge digesters. You remember we have the water treatment, and the activated sludge is separated from the water, and then it goes into the anaerobic digester. And from there, we were taking our samples. So this is then digested sewage sludge. Okay, before we are coming to the sequencing, I wanted to have a basic overview on what I'm dealing with. So I was analyzing a wide range of chemical parameters, like the chemical oxygen demand, organic carbon, nitrogen content, conductivity, dry matter, organic dry matter, pH, and biogas. Well, do you know all of them? Is there one that you don't know? Well, I suppose that you know almost all apart from the COD. Do you all know what a COD is? Chemical oxygen demand. You know it? What is it?
It's the amount of oxygen that you need in the sample to oxidize everything there. So you make a laboratory test with a powerful oxidant, dichromate, potassium dichromate or something like
Yes.
that. So you check how much oxygen it would consume.
That's right. Yeah, that's perfect. So basically it shows us how much oxygen we need to fully oxidize the whole material, and this is also equivalent to the amount of biogas that we could produce. Not always, because there are also non-organic substances which could be oxidized and they will not yield biogas. It could be also that there are substances which are difficult to degrade, like lignin. You could analyze the amount of COD from lignin, but then it will not be degraded in the biogas plant. So it's not the best estimate, but it is still an estimate to say whether there is a lot of biogas potential or not. So I was analyzing all of them, and I wanted to show them in one graph. So I started with many diagrams, and in the end it was really chaotic. So in the end I was just playing around with a program called GNU-plot. You may have heard of it. And I tried to plot every possible combination with always 3 chosen parameters. For example, this one, this one, and this one, or this one, and this one, or this one, this one. And I repeated it until I had plotted every combination. And then I was just overplotting it. So there's an overplot function in GNU-plot where you have an overlay of all the plots that I was creating. So then it creates one big data cloud. And then I was just colorizing the data points according to the plant origin. And I found this quite interesting, that you suddenly see a clustering. So this shows us that these digester sludges are extremely different from each other. So whenever we work with a continuous stirred tank reactor, it's not the same as a sewage plant digester. I think this is really important, because if we think about optimizing anaerobic digesters one day based on microbial knowledge, a strategy that works for a sewage sludge digester might not work for a continuous stirred tank reactor from agriculture, because it has a completely different microbiome. Or at least I was assuming so. Until this point I didn't know. I only know that knew that the sludges were very different based on chemical parameters. So now I had the assumption that also the microbiomes are different, and I was going to analyze this. And here's the first result. This is a typical taxonomic profile. So you see many bars. Here you see a legend. And each bar is one sample that we analyzed from a continuous steered tank reactor or from a plug flow. So basically this type of biogas plant. So what do we see here? What do you see here? Would you make any conclusions based off this figure, or based on this figure?
That Bacteroidetes and Firmicutes are the most abundant bacteria in the reactor.
That's right. Yes, that's right. This was interesting because this was the digestate. So we had several reactors which were really active methane stages, and then we also had samples from an digestate storage. And here the ratio of Bacteroidetes and Firmicutes is really changing. This was quite interesting. But can we do anything with this knowledge? Do you think yes or no? Is it somehow helpful? Well, it's really difficult to make any concrete conclusions from this, because those are really, I mean, this is a classification on a very high level. We are here working at phylum, and you can go 6 levels lower. So this is extremely unspecific. We couldn't do it more specific. And so in each of these groups, we have probably thousands of specific microorganisms. So we see that here is a pattern. And there are many other major groups that you could find which are not shown here. So there is a kind of specificness that we found to this microbiome. But still, there are hundreds and thousands of different microorganisms which are still active here. Okay, here's the next one. This is now the scheme or the chart for the garage reactors. This was interesting. It looks quite different, right? So we also have the blue ones, the Bacteroidetes. We also have the Firmicutes, but not so many anymore. Here we have some Thermotoga. And we have a lot of Spirochetes. Again, it's very difficult to make any concrete conclusions here, because for each group you can have probably thousands of subgroups where we don't know who they are and what they are doing. So it's a rough scheme, a rough pattern again, what we are seeing here. There's maybe one interesting information, these Spirocheta. They are usually known as good swimmers. And this made sense here because we were only accessing the liquids from this garage fermenter. If you remember, we have this piled biomass, and then we have the liquid manure, which is trickled over this pile of biomass in order to always keep it wet, and then it's degraded slowly. So this is how the liquid is composed. And we couldn't access the solids because then they would have to open the whole garage. And this is not so nice to do it in the running process because it's anaerobic, and when they open it, of course, then they lose all the gas and have to get it anaerobic again. So we just said, okay, then we just take the liquid phase. And I could imagine now that the Firmicutes, the green ones here, that they are in much higher abundance in the solids in this pile of biomass. We saw them already here, and very generally the Firmicutes are well known for the degradation of plant biomass. So it's a green biomass, green waste. And so very often they're sitting on the waste, degrading it there. Yes, please.
I'm sorry. I would like to show us the last one slice when we can see all of the combination, pH and etc.
Yes, please.
Okay, and how to make the better decision in order to obtain or get the next result? I guess, is there a lot of combinations that you can make?
I mean, this was just to show the chemical parameters that we have.
Yes.
Just to give a basic overview, we could theoretically analyze each of the parameters separately. This would be more accurate. It would take a lot of time. But in this case, I was only interested in how different the sludges are.
Ah, okay.
You could actually use a principal component analysis as well. Maybe you made this already, you learned how to do it. The principal component is the usual calculation that is done here. But we did it as well, actually. We have in the paper also a principal component analysis. But I included into the main part this and principal component analysis into the supplementary, because this looked much nicer. The principal component analysis is usually very abstract and not so easy to catch. And here's really something easy to understand, and it also gives some additional information. For example, the thick sludge is usually at higher values than the sewage sludge. And the leachate is somehow in between, which also makes sense.
Okay.
So because the thick sludge, I told you, it's so thick that you can almost hold it in your hands, and this is because of a very high concentration of substrate. And in the case of the sewage sludge, it's already pre-digested. It's already degraded in the activation stage. And then once you bring it into the main fermenter, there's not so much chemical oxygen demand left, which you can use for methane production. So these information are given in this figure, but not in a principal component analysis. Okay. So that's why I have chosen to use this one. Although a principal component is definitely more accurate, but it was verifying what we have shown here. So in this regard, it's fine.
Thank you.
You're welcome. Okay, so now let's go to the next one. This is the sewage sludge. Again, it looks really different. So we have also a lot of spirochetes. Makes sense again, because the sewage sludge is relatively thin compared to the thick sludge. We also have the Bacteroidetes here. So apparently they are in all of the plants. They seem to be important for all kind of digesters. And then we have the Chloroflexi. They seem to be exclusively for sewage sludge digesters. When we were writing the article, we were searching for Chloroflexi in other biogas plants, which are not sewage sludge plants, and they can be found, but it's not so common, it's not so often. So I would say it's a rare case, and it seems that the Chloroflexi are really often to be found in the sewage sludge digesters, as well as the Proteobacteria, those ones here. They're not that many, but we found them repeatedly in different kinds of plants, and we also compared this to other literatures, and then we found similar results. So it seems to be a specific case that we have the Proteobacteria in the sewage sludge digesters. Again, there are exceptions. For example, when I searched for Proteobacteria in other biogas plants which are not sewage sludge digesters, I also found some examples, but usually they are not that abundant, and they are rather found in sewage sludge digesters. So if we now look at all of them at once, I found it really interesting that there are clear differences, that we have a more or less specific pattern for each sludge type. So it means there is really a completely different microbiome in each of these sludge types. Please remember that this is relatively inaccurate. If we would go the 6 levels down onto the species level, then we would have thousands of microorganisms, and then probably each biogas plant would be an individuum. So yeah, so these patterns are better to be observed on a rather unspecific taxonomic level. We did the same with methanogens, with the methanogenic archaea. And you may wonder why we did it separately. Why not having all in one graph? What do you think? Why have I done this? So in this case, I was performing one sequencing for all the bacteria. And then I repeated the whole procedure for the methanogens. Why? How could we explain this? No ideas?
Maybe because you want to stop the production of these archaeas. So maybe you want to need who or which archaeas are going to be and whether it be after the process or no, maybe.
But how would I stop them? I mean, you can imagine we took the sample and then we extracted the DNA. The DNA contains archaea and bacteria. So we had all together in one sample. And then we were sequencing bacteria and archaea separately. Or let's change the question. Let's ask it that way. How can we sequence them separately from each other? What can we do differently to have in one reaction just the bacteria and in the other reaction just the archaea? How can we achieve this?
Using specific primers for
Yes.
bacteria and for archaea.
That's absolutely right. So we have chosen primers which are specific for bacteria, and we have chosen primers which are specific for methanogens. So any idea why is this? I didn't explain it yet, so maybe it's a difficult question, but maybe you have an idea. I don't know. I wait because I think they have an idea. No? You can both.
Can you repeat the question, please?
Yeah, why have we chosen different primers? Why did we choose to sequence archaea separated from bacteria? Why not sequence all of them together? I mean, the costs are twice as much. And I told you that it's not that cheap to sequence. So why did we invest so much money to do this?
For most deep in each group, for a major number of sequences of bacteria, analyzing only bacteria. Well, for this.
So to analyze it in greater depth. Yeah, that's actually right. And the reason for this is that we have a different abundance of methanogenic archaea and bacteria. So it could be that you have in one biogas plant just 1% of methanogenic archaea and 99% of bacteria. Sounds strange because methanogenic archaea are the key player which are producing all the methane. But apparently you need in some plants 99% of bacteria to digest all the material until you have enough hydrogen and acetic acid. And then 1% of archaea is in some cases enough to convert all of these acids, the acetic acids and the carbon dioxide and the hydrogen. So, but then if we have, let's say, 1% of methanogenic archaea and 99% of bacteria, it could be that we see almost no methanogenic archaea in our sequences because it's just 1%. And if we don't have too many sequences and then only 1% of them is archaea, well, then it's not really representative. And if you use primers specifically for the methanogens, then you can be sure that you get a lot of sequences for the methanogenic archaea. Okay, now we analyzed the metabolic pathways. Now you might think, how is this possible? Because I told you that with 16S we can also compare taxonomy and that we need to sequence the whole gene to say something about the metabolism. Well, there's a nice database. You know it already. We talked about it in regard to the quorum sensing, in regard to the ability of microorganisms to sense their presence, to sense how many are there. And this map I also showed from KEG, the Kyoto Encyclopedia of Genes and Genomes. And in this case, I was just typing into the search field methane, and then I got all the pathways. And there are not so many. There's the hydrogenotrophic pathway, the acetoclastic pathway, and yeah, here's one which was not that abundant, the methyllotrophic pathway. And there are also not so many methanogenic archaeas, because there's an interesting thing. This was done on genus level. For some reason, we were able to be much more accurate with the methanogenic archaea than with the bacteria. So here we have 6 levels below on the taxonomic ladder, rather unspecific. And here we have just 1 level below on the taxonomic ladder. So this is extremely accurate. So apparently there are not that many methanogens. And if you search for these terms, you can actually find which kind of metabolism they are using. So based on the ratio of methanosacina, for example, or methanosaeta, I was able to know how abundant each of the pathways is. So we had the hydrogenotrophic, the acetoclastic pathway, and then we have something strange here, which is all pathways, and this is related to methanosacina. And methanosacina is a methanogenic archaeon of really high interest in the research society because it can apply all pathways of methanogenesis. I don't know why the other can't do it, but for some reason methanosacina has evolved in such a way that it can use all possible pathways for methanogenesis. It's really versatile, and therefore people believe that it could be a key player in regard to very efficient biogas plants. But this is predicted, or it's a hypothesis, I would say. I don't know whether we can really say that this is true. Well, I mean, at least these 2 plants are high-performance plants, so we can say it's a high-performance methanogen, but we still don't know, at least I have never seen an article that explains why in some cases methanosacina prevails over methanocoleus or the other way around. So in some cases methanocoleus seems to dominate the whole process, although it is less metabolically diverse than methanosacina. This question has not been addressed so deeply into the literature, or at least I didn't find the articles, so to the best of my knowledge this question is still to be solved. So, and well, because of this, methanosacina was really shifting into the focus of my interest, and I was working a lot doing my postdoc thesis on Methanosarcina to understand its role in the microbiome and how we might enrich it and how we could take profit out of it. It's still a hypothesis, but we get more and more evidences that Methanosarcina has some really nice skills that we could use to improve anaerobic digestion. Well, so we are done with this article now, or almost done. And maybe we should talk a little bit about the methanogenesis, because I guess you don't know what hydrogenotrophic means, or acetoclastic, or methylotrophic. Those are the pathways for methanogenesis. I will just go very roughly into these pathways. I mean, you asked already what is coenzyme M. I don't know who asked this. You asked this, yes. So we will not talk about coenzyme M. I had it actually in my slides before, but it's time consuming and relatively long, and I don't want to bother you with so much biochemistry, so I decided to make it a bit more simpler and just go into the basics of methanogenesis. And yeah, really often we just talk about the hydrogenotrophic pathway and the acetoclastic pathway, because the methylotrophic pathway is not that abundant, so we can forget about it now. We will focus just on these two. So it starts with the provision of carbon dioxide and hydrogen, or acetic acid and acetic acid and water. So those are the main substrates for the methanogenic archaea. So if you have actually hydrogen and carbon dioxide, then we have hydrogenotrophic methanogenesis. But if the methanogenic archaea are growing on acetate, then we have acetoclastic methanogenesis. So here you see the formulas, how the archaea are doing it now. Here it looks really simple. So we have 4 hydrogen molecules, then we have 1 carbon dioxide, and we convert it to 3 water and 1 methane. If you would put this into a biochemical route, there are like 10 steps of biochemical conversion how to achieve this. So it's not so easy for the organisms to achieve this. And here we have the acetoclastic methanogens, which are able to directly take up the acetate, and then they make carbon dioxide and methane out of it. So they can basically split it into molecules, and then you have the methane and the carbon dioxide. So what happens if we have a lot of acetoclastic methanogens, but we have a lot of hydrogen and carbon dioxide produced? What would happen to the process? How could we produce methane out of this? We have now carbon dioxide and hydrogen, and they can't consume it. And let's just assume we don't have these guys. So we have just these ones, and bacteria for some reason produce a lot of hydrogen and carbon dioxide. What's going to happen? Any ideas? No one has an idea? You're shaking the head. Okay, I will ask differently. Who thinks that methane is still produced in such a scenario? You can just raise your hand if you believe so. So you are all believing that no methane is produced anymore?
I think it depends of the archaea, as the other slide. If there is the archaea that can use different paths, maybe instead of using acetoclastic, it will change to hydrogenophilic. So it would be certain production of methane, but not as much as.
Okay, if I understood you right, you mean that that there are methanogens which can change their way of metabolizing it.
Maybe.
Okay, well, this would be possible for metanosatsina. Yeah, I agree. Let's assume we have here methanotrix or methanotrix, which is only able to eat acetate. It's not able to consume hydrogen and carbon dioxide. But then we're producing a lot of hydrogen and carbon dioxide. What's going to happen?
The reactor is going to acidize.
Into acidosis?
Acidosis, and the methane production is going to be
Inhibited, yeah.
Yeah, that's right.
Could be one option. Well, we have these guys, and if we are lucky, then they could convert the carbon dioxide and hydrogen to acetate. So you have basically the products for the methanogens, but depending on their concentrations, they can bidirectionally be converted, and this of course depends on the chemical equilibrium. So if we have a lot of acetoclastic methanogens and we produce mainly carbon dioxide and hydrogen, then it's very likely that they consume all of the acetate. There's not much left. So, but then we have a really high gradient from there to there. So it could be energetically quite favorable for the homoacetogens to convert the carbon dioxide with the hydrogen into acetate. And then again, they have enough to eat. So there is some diversity to adapt to such stressful conditions in the microbiome. It's another example why we can say that the microbiome is really robust. Okay, now we also understand a little bit better this year. We now have an idea what these pathways are. Well, and what's the conclusions? So we have seen very specific patterns for each of these microbiomes. But I told you about the taxonomic ladder. And in the case of archaea, we have been relatively specific. So we could climb one ladder step higher. So this is really, really accurate. But then with bacteria, we somehow remained here. I don't know exactly the problem, because I'm not a bioinformatician. I could imagine that it's because we have so many different bacteria, so it's more difficult to really compare them, to really distinguish them with high accuracy. Maybe also many of them are not known, so there are still many unknown bacteria inside the process. Yeah, so those are the reasons that I could imagine. But if you're talking to a bioinformatician, he might give you further reasons why we could only achieve the phylum level on a bacterial level. Well, there are methods to still achieve this, and they are more expensive. For example, you could sequence the whole metagenome, the whole DNA sequence. And if you do so, then the few phylums that we observed here, they will explode in their number, and then we have a huge number of species, which is really hard to oversee. There's an interesting article. It's not too old. The impact of immigration on microbial community composition in full-scale anaerobic digester plants. And they wanted to analyze the core microbiome. And they found that there are 300 O2s. What's an O2? Has anyone heard O2 before? Stands for Operational Taxonomic Unit. And an operational taxonomic unit refers to sequences which have a similarity of 97%. And you apply this concept when you don't know the microorganisms. Because you have these databases in green genes or silver, and then usually you have many organisms which you can't classify. So in order to still work with them, you just search all the sequences which are really close to each other, and you define them with one O2, and you give them a number, O2 1000. And then if you reanalyze them, you already know, okay, that's the number I was working with. So you have a possibility to distinguish them somehow. Well, we have now these 300 O2s, so we could simplify it and we could say we have 300 species, which are forming 80% of the biosynosis of all the reactors they analyzed. And they analyzed, I don't know whether it's set here, I think it was 21. No, they don't say it, but I think it was 21. So they analyzed quite some reactors, and yeah, so it was interesting that there is something like a core microbiome, and 300 might sound a lot to you. I can't definitely not remember 300 taxonomic names of bacteria. So for me, those are a lot. But if we take into account the 20% which are not in the core microbiome, and which are not that abundant, and if we take or sum up all the ones from all the digester they analyzed, then you end up with thousands of different microorganisms. So you have a huge diversity, but then a rather small core microbiome. So what's behind these O2s? People are continuously working on this. So basically they try to find a DNA from a specific microorganism, and then you have to sequence the whole DNA. Not just one specific gene, but you really have to sequence all the DNA, at least if you can't culture it. So the best way is still to culture a certain microorganism, and then you can give it a name, you can publish the name, and that's it. You can even give it your name if you want, so as you wish. Well, that's more difficult on a sequence level, but still we have the possibility to search like a puzzle all the DNA which corresponds, for example, just to this guy. So the bioinformaticians are puzzling all the DNA parts together until they have a closed circular genome, and then they know, okay, this must be this guy, and then we can check what genes he has, and from this we can conclude what he might do, although we can't culture it. So this is really nice. And we did it, not I personally. I was collaborating here with some bioinformaticians. I sent them some interesting samples from biogas reactors, and then they were able to sequence all of them, and we were able to publish this. So we found, for example, a new genus for Clostridium, or a new Bacteroidaceae, it's a new family, or a new Osyrospiraceae. Yeah, this is also on a family level. So this was quite nice, because if we just take this, taxonomic ladder, this is more or less here. And it's quite common that people are describing new species all the time. So this happens quite often. But it's more difficult to find a new family or a new order, or even a new phylum. If you find a new phylum, this is already quite amazing. But a new family is already something. And we did it here, so this indicates how much is there still unknown, how much we still have to learn to fully understand anaerobic microbiomes. Well, this is, for example, one genome that we published here. Those are usually relatively short articles of 2 or 3 pages, where we just give the DNA sequences and then you write some metabolic highlights about it. So what could we do now with this? I told you already we could get some idea about metabolic capabilities. So in the end, it's a work from bioinformaticians for bioinformaticians. So for us, it's not so helpful in the end. But the bioinformatician could, for example, create such a circular map with it. So you have all the DNA from your bacterium, and then they can assign some names for proteins and enzymes that are to be found there. And then we could go to the Kyoto Encyclopedia of Genes and Genomes and could check what they are doing. Then we could get an opinion what it's doing. So with this, we can do something even if we are not a bioinformatician. This is another work which highlights the huge amount of organisms that we don't know yet. So here it's a big group, and they did exclusively bioinformatic work. So whenever someone is sequencing something and he wants to publish it, he has to load it into a public available database, like the NCBI database. So for example, the sequences for this will be somewhere on a database, and you can download them and you could work with it if you want. So I told you we have already hundreds or even thousands of articles on metagenomics. And these guys, they were just downloading all of it. Then they had a huge amount of sequences. And then they were trying to puzzle it together. They were searching for all the sequences which are slightly overlapping. And then they were constructing genomes from this huge soup of DNA sequences, let's say. And they were able, by doing this, to find 1,600 species from an anaerobic digester. So this is amazing. And again, this highlights we still have some work ahead of us to fully resolve this anaerobic microbiome. Many people therefore still refer it as a black box. And well, I hope that in not too much time we will be able to resolve this black box. And this brings us also to the term big data. At the moment, big data is quite nice funded at the European level, for example, because in order to work with such complexities, we need completely new mathematical and bioinformatical approaches to handle this. So what is a metagenome? So far we learned about the 16S gene, and now how should we sequence the complete genome of something? Where do you start? Which probes can you use for this? So here's a scheme. It's a bit small, but I can explain it. So we have our DNA, and then we are fragmenting it. So you have some enzymes or some physical forces. So there are ways to share the DNA in smaller parts. And then you basically sequence all of them with randomized primers. So you know already what a primer is. And there's a way to have a mix with all kind random fused nucleotides. So you have a huge amount of different primers. Well, they're attaching somewhere on these fragments, and then you sequence basically everything. And once you have sequenced everything, you really puzzle it together like this. Of course, if you're a bioinformatician, you can apply some algorithm to find the overlaps much faster. And then in the end, you get a sequence like this. And if you're lucky, you can close your genome. And then you have the full genome for a new organism that wasn't known before. Well, I think we have now a relatively good idea of the differences of the different reactor types, their microbiomes. Now we know the different pathways. There's an interesting thing about the direct interspecies electron transfer. So apparently these guys and these guys can use something else. People maybe don't call it a metabolism, but they can use this direct interspecies electron transfer to perform these pathways. And this has a huge impact, which we will also discuss later in more detail. And now I made like a short comic to highlight or to explain how these shifts can happen, to get a deeper understanding why we have those ones here, and why we have those ones here. So let's have a simple case in sewage sludge digestion, for example. We will form some acetate. Usually at lower concentrations of biomass, there is not too much hydrogen, there is more acetate. And all this is consumed by Metanocyta, which meanwhile has renamed to Metanotrix. So Metanotrix is able to do this. It's a really simple scheme. We have the acetate and it goes into the metanogens, and that's it. This is possible until, let's say, 3,000 milligrams per liter. And then something is about to change. Suddenly, more and more hydrogen comes up. So apparently, the organisms are not able to convert it so efficiently to acetate, or based on the chemical equilibrium, more hydrogen is given. So we have just hydrogen and acetic acid in our digester. But now we have a problem, because we have Metanotrix, or Metanocytina, which is only able to consume acetate. We heard before that it's possible to convert the acetate into hydrogen, but if the concentration is too high, well, this does not help us too much. We will still produce hydrogen, and it's not consumed. So what are we doing now? So, for the moment, nothing. They just can't eat it. And suddenly, propionate and butyrate is accumulating. Why is that? So, usually we have this oxidation of organic acids, which happens step by step. So, they are getting shorter and shorter until you're left with the acetate. In every round of digestion, the so-called beta oxidation, you split 2 C atoms from your organic acid until you remain with the acetate. This is dependent on the hydrogen, and usually this happens in a syntrophic reaction. We learned already what syntrophy means. It means interdependence in metabolic collaboration. And these acetic, syntrophic acid-degrading microorganisms usually need to get rid of the hydrogen. The hydrogen is inhibiting them. And if there is no one who is consuming the hydrogen, and the acetoclastic ones can't consume the hydrogen, then the hydrogen will enrich. It will inhibit our syntrophic acid-degrading bacteria, so they can't degrade the propionate and the butyrate anymore. Suddenly, we have a more complex spectrum of organic acids, and that's something which we can measure really easy. So if we are supervising a biogas plant and you measure large amounts of propionate or butyrate, you know something is wrong, so better stop feeding. So this is really a nice indicator that we can use here to improve anaerobic digestion processes. But still we have the problem now. I mean, we could stop feeding. This would probably work. But maybe we say, well, we want to have a more efficient biogas plant, so we have to get the hydrogen somehow into methane. So how can we achieve this now? Here I also gave the concentration, the pressure at which the hydrogen becomes toxic by 10 to the power of minus bar. It's so toxic that a reactor will very likely undergo acidosis. It's also interesting, I mean, the reactor is now full of substrate, but the acetoclastic bacteria are mainly starving. They could probably eat more, but there is not, it's just not enough acetic acid to consume. Instead, there is butyric acid, propionic acid, and hydrogen. So there are more and more substrates suddenly accumulating, which can't be consumed directly by the methanotrix. So suddenly the hydrogenotrophic archaea come into play. So these are now the optimal conditions for them to grow, and they can directly consume the hydrogen. And when they do so, the pressure goes down. We are not inhibiting the propionate and butyrate degrading syntrophs anymore. So then if everything works right, we don't have propionic acid and we don't have butyric acid anymore. Instead, we have more acetic acid again and hydrogen, and the reactor is again able to work. Yeah, there's a very specific range at which we can work here. So usually between 3,000 and 15,000 milligram acetate per liter, we are counting to have more hydrogenotrophic archaea, and usually the acetoclastic ones are suppressed at this range of acetate. Okay, I think now we understand this even better than before. But there's still one question. Why do we have at very low concentrations the metanocyta or the methanotrix, but not the metanosatsina? Both of them are able to degrade acetate. So why can't metanosatsina be here as well? And this is explained based on our chemistry. There's an enzyme called acetate kinase. And the acetate kinase is extremely efficient for methanotrix, but it has a very low turnover rate. So due to its affinity, it can catch the acetate really efficiently, even at very low concentrations, to convert it into methane. But if the concentration of acetate is very high, it can't convert it very fast. So it can bind the acetate very efficient, but very slow, and it's turning it very slow into methane. So therefore, the whole process will be very slow. And metanosatsina has the opposite case. Metanosatsina also has an enzyme to take up the acetate, but it has a low affinity with a high turnover rate. This means when there is low amount of acetate, it just can't bind the acetate, so it's starving. But when the concentration gets a bit higher, then suddenly it can bind the acetate, and then it turns the acetate much, much faster than the methanotrix could do it. So this is just an explanation why we have the metanosatsina here in this high-performance plants and the methanotrix in these low-performance plants. Just check the time, see how much time we have left. Okay, we have still half an hour. Is this more or less clear? You're nodding, great. The other ones? What did you not understand? Did you understand? Okay, what is difficult for you?
Yes, I have problems with microorganisms to understand.
Yeah, okay.
So, but I guess that I got the point.
You got the concept, okay.
Yeah.
Great. Okay, yeah, here's the turnover rate again. I should have clicked 1 point further. Now we have a really interesting case. So imagine that we just provide carbon dioxide and hydrogen. This is the perfect substrate for our heterogenotrophic methanogens. And actually, in such a scenario, they can work much more efficient than before because we're not feeding biomass. If we would feed now biomass, all the hydrogen would inhibit our syntrophic bacteria, which are degrading the acids. So we would go very fast into acidosis, and the whole process would die. But here, we are not feeding biomass, so there will be no formation of organic acids. We are providing separately the hydrogen and the carbon dioxide, and suddenly the methanogenic archaea can grow much, much faster because we can have much higher substrate concentrations for the methanogens without inhibiting the syntrophic bacteria. So therefore, these kind of plants, we call them power-to-gas plants, are extremely fast. And there are different concepts how we can use them. So we distinguish between the ex situ process, the in situ process, and the hybrid process. All of them have the same goal because sometimes we have too much energy produced, and this especially concerns us now as we try to use more and more renewable energies. Because the fossil fuels, we can produce them on demand whenever we want them, but when the wind is blowing and the sun is shining, the energy is coming, and then we have to do something with it. And one option is that we perform electrolysis. I think I explained this also in the very first lecture, maybe you remember slightly. So during electrolysis, we produce oxygen. This is nice. Oxygen is always good. And then we have hydrogen, and we have to do something with it. We can't store it very good. It's possible, but expensive. We don't want to put it in the gas grid because the gas grid is made for methane, for natural gas. And also, if you compare the volume of an equimolar mass of methane to hydrogen, the hydrogen will have a much larger volume. So if you have 1 methane, you have 1 mole of methane, which is equivalent to 2 mole of hydrogen. Yeah, that's right. So this means you have a much larger volume which you put into the gas grid. So in this case, you would have much less potential to store gas in the gas grid. Also, it's dangerous to have hydrogen into the gas grid. For example, if the gas grid is approaching fuel stations, it's mandatory at the moment to have low hydrogen concentrations in the gas, which is coming out of the gas grid. So therefore, it would make sense to somehow turn this hydrogen into methane, and we can do this if we feed it now into the reactors, but we need carbon dioxide. And carbon dioxide is produced in biogas plants. Therefore, it's really nice to combine power to gas with a typical codigester. For example, in an ex situ process. Here, we are producing out of biomass a biogas which contains mainly 50% of carbon dioxide and 50% of methane. So now you could feed this gas into your power-to-gas reactor, and you could supplement the hydrogen from the electrolysis, and then the hydrogen could be used to chemically reduce the residual carbon dioxide from your biogas, and suddenly you would get a huge, or a really enriched form of your biogas. With this, it is possible to reach methane concentrations up to 97%, and that's the requirement for the gas grid. So this is actually a really nice upgrade technology which makes it possible at all to give this gas into the gas grid. But you could also think about pouring the hydrogen directly into the biogas plant to upgrade your biogas. This is the so-called in situ process. And the in situ process has a very big problem. Do you know which? What would happen? No one has an idea? What would happen if we take a co-digester, which is fed with biomass, and suddenly we pour in a lot of hydrogen? No one has an idea? Do you remember this inhibitory effect for the syntrophic bacteria? So we can go back a little bit.
Yeah, the acidification of the reactor.
Yes?
Because there's a lot of concentration of hydrogen, so
Yes.
well, archaeas can't produce methane.
Yeah, you go into the right direction, but the ones which are inhibited are not the methanogenic archaea, but the syntrophic bacteria which are degrading the acid.
Yes.
So I don't know whether...
Sorry, so they cannot produce acetate?
Exactly.
Okay, yeah.
So usually you have this syntrophic bacteria, and they can degrade butyric acid and propionic acid. But at this point, they have to get rid of some hydrogen, which is usually consumed by methanogenic archaea. And therefore, you have usually a very small concentration of hydrogen, which still enables the syntrophic bacteria to degrade these organic acids. But then, if we suddenly feed in some hydrogen, the level goes so high that the syntrophic bacteria are inhibited. They won't degrade the butyric and the propionic acid anymore. The pH drops, and then the methanogenic archaea are inhibited as well. So it's like a chain reaction in the inhibition. But as long as we don't need syntrophic bacteria, because we are only feeding, in this case, only carbon dioxide and hydrogen without biomass, we don't care about syntrophic bacteria. So in this case, we can feed as much as we want, and the methanogens are happy to receive as much as possible. Well, and then there is this hybrid process where we just try to have maybe partially upgrading of a normal biogas plant together with a separate power-to-gas reactor. But I would say this is the best option, because here you can be really sure that there is no inhibition at all. Oh, I already asked this question. So how do hydrogenotrophs ferment acetic acid? So we answered this question already. Because we have the syntrophic acetate oxidizing bacteria. Do you still remember them? Yeah? I can show you the picture where we saw them first. I mean, we don't saw the bacteria, but we saw what they are doing. This picture. Here we have the syntrophic acetate oxidizing bacteria. And if we have too much acetate but no hydrogen, they can turn it and make it accessible. Yeah, so this would be possible. We have our acetate. We turn it in hydrogen and carbon dioxide due to our syntrophic acetate oxidizing bacteria. And following this, our hydrogenotrophic bacteria can also eat it. Well, and the best thing is if we have methanosarcina, the one where we set all pathways, this really versatile or metabolically versatile methanogen, which can perform hydrogenotrophic and acetoclastic methanogenesis. So in this case, we don't care whether we have hydrogen or acetate. They can just consume everything. And we can even accelerate them if we just throw in some conductive particles. They can now sit on these particles in order to perform their pathways even faster. Not all methanogens can do this. For example, methanoculeus, this purely hydrogenotrophic methanogen, can't perform this electrofermentation. But methanosacina can, which makes it, again, a really interesting methanogen. And it really sits on these conductive particles in order to use them to boost up its pathways. Here's one article which has shown this. They were adding 20 grams per liter of red mud. That's a residue from the mining industry. And with this red mud, which is usually toxic, but in this case, the methane production was increased very much, 35%. It's a lot. So you see that due to this electroactivism, you could really achieve something. And this now becomes really interesting for the industry and brings us to the next topic, MIET and DIET, which stands for mediated interspecies electron transfer and direct interspecies electron transfer. Okay, we also have some time left, 15 minutes, so we can at least get started here. There's a really interesting article, and they were working with a crystal called phenazine. I was wondering why they were choosing the phenazine, but then I saw that there is a relatively similar substance in the metabolism of methanogens. So I think this was the idea, just to choose this phenazine. And they were forming some crystals out of it. So this was some chemical work. And then they were putting it into an anaerobic digester. And then they saw this. So the green dots are methanogenic archaea. Why are they green and which method we are using? Do you remember this method? We talked about it in the third lecture when it comes to methods that are suitable for analyzing anaerobic microbiomes. There was one particular method which gives results like these ones.
It was fluorescence?
Yes, it was fluorescence. And what was special here? It was a very specific kind of fluorescence microscopy. Which one do you know? No ideas? Okay, so I will resolve it. You could use the fish microscopy, the fluorescent in situ hybridization. This is possible, this would work, but I would not recommend it because it's expensive. You need these probes, the protocol is very long, it's quite some effort. We have the DAPI, but with the DAPI we see everything. We see methanogens, we see also the bacteria. So again, we need something else. So then there was a third method that we talked about, and this was the F420 microscopy. If you remember this, the F420 was an electron carrier, which is used specifically by methanogenic archaea, at least under anaerobic conditions. And this metabolite is able to be fluorescent if you use the right wavelengths. So you don't have to use any fluorescent probe, no dyes. You can just apply the right wavelengths, and then the methanogens will directly start glowing with a green color. Okay, now we see all the methanogens here, and then we see these fibers in between. And you see that these fibers, those reddish phenazine crystals, I was amazed by them. I mean, it's also amazing to see that how flexible these crystals are. And all the methanogens are now attaching to these fibers. So apparently they really enjoy this conductive surface, and this phenazine is conductive. So they like to grow on conductive surfaces in order to be faster, to work better. It just gives them a better environment. Why is that? So here's a simple scheme on what MIT and deity actually means. So I told you that electrons are kind of waste at some point if we don't know how to retrieve energy from it anymore. So at some point we have taken everything we can, and then we have to get rid of the electrons. And one way is that we just make it react with water at our electron transport chain, with oxygen, sorry, to form water, the electron transport chain. But then there might be some organisms which would use it not fully, oxidize it fully, and then there might be other microorganisms which are able to further oxidize the electrons to get some energy out of this. And this could, for example, be the case with Geobacter and Methanosarcina. So Geobacter could take a tiny bit of energy from the electron, and then it could send it to Methanosarcina, and Methanosarcina takes the rest. So like this, Methanosarcina has something to eat, and Geobacter is happy that it gets rid of its electrons, which it can't use anymore. So this transport can happen in different ways. So one way is this mediated interspecies electron transfer. We also talk about IIET, which is very similar, the indirect interspecies electron transfer. And this always happens with some carrying molecule, for example, acetate or hydrogen. Yeah, so in the case of Geobacter, it might not be able to eat or to retrieve energy from electrons that are packed in acetate. So it tries to get rid of the acetate, and then the Methanosarcina can metabolize it further. But now there's another way. Instead of sending the electron packed in hydrogen or acetate, you could try to move it more directly to the methanogen using these kind of conductive fibers we talked about, these nanowires. So they're electrically connected, and like this they're sending the electrons to each other. But then there are some ions remaining. For example, in the case of hydrogen, you would have protons and electrons. The electrons are just transferred via these fibers, these nanowires. The protons are going into the medium that surrounds the organism. And the protons are wandering relatively fast, so they can be taken up very fast in order to produce hydrogen again in the cell to perform then the hydrogenotrophic pathway. Yeah, and that's basically it. So in the MIET, we need a carrier, and in the diet, we don't need a carrier. And then it could look like this. That's the pictures that you probably remember. This can be shown in different ways. And one way is to use an electrical cell with an anode and a cathode. And on this anode, you could, for example, grow Geobacter. And on the cathode, you could grow Methanosarcina. You could even have a membrane between it to be sure that they are not exchanging between each other, to be sure that you have really just Geobacter here and Methanosarcina here. And then Geobacter wants to give its electrons to Methanosarcina, and since they're not directly connected, at least not in the medium, the only way to achieve this is to send the electrons along the anode, and once they're approaching the cathode, they can be used by the methanogen. And this can be measured. So with this, we can actually measure that they have a way of electrical communication. So here you see a scheme from all three possible ways that we distinguish here. I mean, we have the MIET and the DIET, but the DIET can still be distinguished in two different ways of how diet is happening. So the upper one is the MIET. Here we have our organism which can't grow any further on hydrogen or format or any other metabolite, and then it's diffusing to our methanogens, and they are now making methane out of it. So that's the MIET. Now we have here the diet, and here we have our nano wires, these electrical fibers, and now the electrons are sent directly. This means the hydrogen is kind of degraded into electrons and protons. Electrons are running here to the methanogens, and the protons are diffusing alongside the medium. Well, and then we have this last case, because some organisms are able to send their electrons directly, but they don't have these fibers, these nanowires. And in this case, they can just sit on conductive materials like biochar or magnetite, and then they can send the electrons via these conductive materials to the methanogens. And this is something that we can use in the industry. So there are already companies which try to sell conductive materials to industrial biogas plants and recommend them to throw it in large amounts in order to increase the degradation rate, to make them faster, to produce the biogas much faster. Is this more or less clear? Okay. This has also another advantage. I mean, it's more direct, but you also prevent a back reaction. I told you that the syntrophic bacteria are easily inhibited by hydrogen. So when too much hydrogen is released, the hydrogen could cause something like a chemical back reaction, and then they won't degrade the organic acids any further. But in this case, they would not even release the hydrogen, because the hydrogen is splitted into electrons and protons, and when they have protons around them, this does not allow a back reaction because the electron sits already here. So with this, you can really improve the robustness and the efficiency of syntrophic bacteria, syntrophic acid degrading bacteria. Is this clear? Okay, great. So if you have any questions, just ask. Yeah, so for a long time, people thought that at least in biogas plants, this MIET is the only possible way. So we don't know about this for so long time, approximately since 2005. Since 2005, we're doing research on this. And the first ones which were able to, I mean, now this can be performed by all the methanogens, hydrogenotrophic or acetoclastic. They all need to get rid of, or they all need to take up the electrons somehow. But now we have the diet, and for a long time people thought that this is, well, not long time, but for some time people thought that this is restricted to some acetoclastics, methanotrix and methanosacina. At least methanocoleus can't use it. But then this view changed, and there was recently an article, well, it's not too recently anymore, 2019, from Walker et al., and they found a Methanosperillum hungatae, which is hydrogenotrophic and also able to perform this diet. Yeah, that's a pity, because I thought it's a nice method to specifically enrich methanosacina. I thought only acetoclastic ones could, I mean, it's an acetoclastic ones, and I hoped to enrich methanosacina by adding conductive material. And, well, methanosacina could be favored because of this, but it's not the only one, so we could also favor Methanosperillum, for example. But it could, for example, help us to suppress Methanocoleus, and I told you that we still don't really understand why we have in some cases a high proportion of Methanocoleus and in some cases a high proportion of Methanosacina. So this is still to be figured out. And this is just a picture which shows you these fibers, these conductive wires. they don't look so special. I mean, it's like a fiber which is empty in the middle, but the electrons are transported on the outer part. So basically the electrons, which are the proteins are sitting in the wall, are so chosen in such a way that the most conductive amino acids are used for constructing these fibers by the microorganisms, and due to this choice of the right amino acids, then they are conductive. Yeah, here's a possible way how this could be now integrated into an anaerobic digester. How this could look like, because usually you have several pathways that are happening in parallel. And we could have here some hydrolyzing, acidifying bacteria, which are releasing organic acids. And then we have the syntrophic bacteria. I told you the syntrophic bacteria, they need really close collaboration with the methanogens in order to prevent that hydrogen is enriched in their closeness. And well, now they have the possibility to make the classical hydrogenotrophic way. This means carbon dioxide and hydrogen, they should have shown the hydrogen here, this is missing, goes both to the hydrogenotrophic methanogens, and they do the classical hydrogenotrophic pathway. Or they could also release acetate, either the syntrophic ones or the hydrolytic ones, and then the acetate goes to our acetoclastic methanogens like metanosaeta or metanosatsina. But now we could also have the case that they are releasing the electrons and the protons separately. So this is basically the product which is used for the hydrogenotrophic methanogenesis, just split it in electrons and protons. But then the interesting thing now is that they can direct the electrons to the acetoclastic ones. So suddenly, metanosaeta can grow on carbon dioxide, electrons, and protons, although it's not an hydrogenotrophic methanogen. So this is quite confusing, but it's happening. Could you follow me on this? Yeah? Okay, great. Yeah, and then we have here the diet. Here we have the MIET. And here I call this HIT, which is also a form of MIET, and it stands for hydrogen interspecies electron transfer. So it's just a more specific form of the MIET. So now we could try to integrate this in our pathways into the hydrogenotrophic and in the acetoclastic ones. So we have the MIET, which we know already very well. And then we could here assign the diet, which sends then the electrons and the protons into this metanosatsina. Well, and since recently, since this work from Walker et al., we know that also the opposite could be possible. So the electrons and protons could also go from here to here. So I still have to correct this. Well, I think the time is over, right? Yes, so we are already over the time. So we will continue here tomorrow unless you have any specific questions. Anything unclear? Oh no, tomorrow, no, weekend. Tuesday, Monday is holiday, so we see you on Tuesday. Okay, then thank you very much and see you next week.